Hello friends. I think in the last session we have completed with general understanding about androecium and gynoecium, that is male reproductive structure and female reproductive structure. We have also gone through the section and observation of a section of ovary. So I think understanding the general structure of any flower reproductive structure and understanding the actual arrangement of ovules or arrangement of any some organ in a given section is a fantastic thing. And now here you can observe there are so many diagrams and so many labels and terminologies are there on the board. And yes, we are going to we will try to recall the things because these things are there in your regular 8, 9, 10 curriculum. So I think you people will really enjoy this particular session because we are going to revise so many things, and yes, we are going to correlate these events, the events which are there with plant's life with that of animal's life. So reproduction in flowering plants, reproduction in angiosperms is one of the most studied branch, and yes, you people are always keen to know what type of careers we can have, what type of branches we can select, what specialization can be done once we select particular branch. So these are the branches which we are studying right now. Once we start with general characterization of plant and naming and nomenclature, that becomes a taxonomy. Taxonomy includes so many things, right from field work to the referencing. There are so many things which can be included. Once you start with studying how structures are involved in reproduction and what are the actual structures which are taking part in reproduction and produces a new seed. Before that, obviously, zygote and embryo. So that part falls under embryology. So this is how any one of you can be a good botanist, good embryologist, plant breeder. So there are so many n number of branches. So always remember, if you think about pursuing any career with respect to plant sciences or animal sciences, you have tremendous scope. So don't stick to a particular branch that okay, I have opted biology for standard 11 and 12, then I must go to medical sciences. So it is not a compulsion. It is not a compulsion by government. It is not a compulsion by any teacher. So it's a compulsion by you people. Instead of that, you should make yourself aware that yes, I'm going to learn biology. I'm going to learn some principles, and these principles will definitely help me to understand how nature works, how organism works. Okay, let there be a plant body or animal body, but overall 99% the principles are the same. If we think about say excretion process in case of humans, similar type of process is operative in plant body. Whatever the mode or pathway for exact excretion is different in plants and animals, but the bottom line remains the same. That is removal of waste material. So this bottom line we need to understand. This difference or this similarity we need to understand by comparative study. Comparative study will always help you to take a step towards success or your goal. Okay. Here we can start actually with the angiospermic reproductive structures. Okay, shall we? Let us start with the board one. So let us revise the things. So this is diagrammatic or schematic, you can say. It's a schematic structure of any flower. This is a dissected structure, you can say. You can easily observe this is a stalk or pedicel. This is thalamus. Then these are the petals, white color chalk. It is petal. Then this male reproductive structure, androecium, which is made up of anther lobe and filament. Simultaneously, female reproductive structure, this is gynoecium with stigma, style and ovary contains ovules. So this basic structure we have studied and you are well aware about this. Let us magnify the things. First we will magnify the anther lobes. So this is anther lobe and I can take a section. So once I have taken a section, the surface will appear like this. It's a top view and I have tried to put the things in a 3D manner. So you can easily observe there are 4 chambers and definitely there are some developing pollen grains. Pollen grains, they are produced in a locule-like structure or a chamber-like structure by the process of meiosis. Simply listen to the terminologies, listen to the concepts. Don't try to get jumbled with or don't get afraid whether I'll be able to remember these things or not. You need to understand first. You will remember everything, I assure you. So this particular structure, this is locule, which is filled with number of pollen grains, and yes, pollen grains after maturity and after dehiscence of anther lobes, so pollen grains will come out and somehow they will appear in this form. So this is a typical representative picture of any pollen grain. I am not saying that each and every pollen grain will appear spiny. There are smooth pollen grains. I cannot say that each and every pollen grain will appear spherical in shape. There are triangular pollen grains. So there are so many n number of varieties which are there in the nature, and definitely these pollen grains, they require some agency to get transferred from one flower to another flower just to carry out cross-pollination. Cross-pollination, always remember, cross-pollination is always responsible for producing new genetic combinations, because pollen grains of one flower, if they are getting delivered to the stigma of another flower, then definitely there will be great chances of recombination, and this is how the I cannot say new variety will be produced, but improved variety, improved generation will be produced, improved offspring will be produced. So this is the major benefit of cross-pollination. So this is how pollen grain appears. So this much we are considering. We are not going to consider the entire development of male gametophyte in this particular sequence. Okay. Let us think about the magnified view of female reproductive structure. So this is stigma, style and ovary. Now this particular section I have taken. So this is a typical section where you can observe a typical ovary wall. Always remember, this particular structure appears three-dimensional in actual appearance. So this is VS, vertical section, VS of ovary. So this is vertical section, hence this part is removed and now you can observe the ovules which are there in the ovary. So this is ovule, and ovule is attached to the ovary wall with the help of stalk. Let us magnify one of them. Let us magnify one of them, and once again this is ovary wall and this particular structure is ovule. Forget about the details of the ovule. Simply think about how this structure is beautifully attached to the wall of ovary. Let me tell you in the beginning itself, deliberately I have drawn this particular structure in a swan neck fashion. So this is just to tell you that ovule is attached to the surface. I can show it like this. I can show it directly like this. I can put it like this. So there are so many combinations, there are so many structures are available about attachment of ovule to the wall of ovary. Okay. So remember, this is a diagrammatic representation. So this is all about how the things appear actually in a plant body. Let us think about development of pollen grain. Or we can say in details we can learn about how pollen grain appears, or how male gametophytes are or male gametes are produced. Okay, so here we can actually rethink about liberation of pollen grain. So this is pollen grain, which appears to be quite harder because there are two major layers, exine and intine. Exine is quite harder, and there are certain records in various research papers as well as certain departments in the world that pollen grains are the typical evidence-like things which can be used in oil extraction units or oil extraction fields just to record that particular oil when it was formed. It is a simple technique. Once the pollen grains are collected from the soil strata, the lowest soil strata, then just because of carbon dating, so it is possible for researchers just to determine what is the exact age of pollen grain. So when the oil traces are found nearby the pollen grain, that time you can say this particular oil strata or oil column was formed in that particular era. So this is how the actual commercial application of this knowledge is possible. So this is pollen grain which is hard one, and even it can remain there with the soil for a longer time. And there are so many cases where you can find pollen impressions. Pollen impressions means there are so many fossil structures which show presence of pollen grains. So this is possible, and yes, these records are available. Let me tell you with respect to this particular additional knowledge that don't Google each and everything which is mentioned here. So do not try to Google everything, because it will create a chaos of knowledge. Knowledge is important. Information will do, but too much of information will cause a chaos. Okay, just to avoid, simply focus on what the structures are being mentioned here. So this is a typical exine which appears to be spiny by nature. Let us think about, this is the structure only. So I have drawn specifically intine only. Now just to show you how things appear, I am deliberately drawing this exine in front of you. So this is exine, this is intine, and in this particular spherical structure, the tube cell and generative cell is present. So tube cell can be called as vegetative cell. So this particular cell is vegetative cell. This particular vegetative cell and generative cell are bathed with the cytoplasm. So cytoplasm is present, and tube cell always responsible for forming a tube, pollen tube, which can be inserted into the ovule just to carry out fertilization. Generative cell, it divides mitotically to form a typical male gametes. So there are two male gametes. That story we are going to learn. So these two male gametes, so they travel in a pollen tube, which is formed by a tube cell. Okay. So let us think about the development of pollen grain, or how that pollen grain grows further. So let me tell you, there are certain agencies. Let there be insects, let there be any bird, let there be any butterfly or water. When pollen grains are liberated on a stigma disc, as I mentioned in the previous lecture, stigma disc is supplied with a sucrose solution. Sucrose solution, it always helps that particular pollen grain to form a tube. How it helps? Pollen grain, it allows that sucrose solution to enter. There are so many, or not so many actually, there are germ pores available. So there are germ pores available through which entry of sugar solution is possible. So this is how the pollen grain becomes turgid. So once that pollen grain becomes turgid, so this particular tube nucleus of a tube cell, so it travels first, and the intine, whatever the intine is there, so this is intine, so it floods out, it forms a tube. You can consider a football outer hexagonal jacket. You are well aware, it's a strong leather or strong artificial material. Okay, then inside the strong artificial hexagonal structure there is a tube, rubber tube present, which is quite smooth, and you can say simply if you put a nail on it, so it will burst out. So this is how the things are there in the nature itself. So consider this as a inner tube, which is a rubber tube, and just because of increased volume of cytoplasm, because the sugar solution which was there on the stigma disc, now it has entered, right? Now that particular increased cytoplasm will try to push the increased volume out. It cannot burst out. It's a slow process actually. So this is how it will try to push out the overall volume of a cytoplasm, increased volume of a cytoplasm, and in that process the tube nucleus will travel first. What is the monitor? So this leader will be there, and so it will travel first. In the meanwhile, this generative cell will divide mitotically. We are not going to study the things in much detail, because these things are going to be there in your standard 12, very first chapter of your standard 12. So we are going to learn all these events in that particular chapter, and before that, even in case of morphology chapter in the standard 11, actual standard 11, we are going to learn some of the events of this particular pollen grain. So this is how the generative cell, it divides into two to form male gametes. So the two male gametes you can easily observe. The blue colored male gametes are present in a tube. So this tube is made up of intine. So this tube is made up of intine. Remember, always remember. Now this particular structure, it's a tube-like structure. Let me tell you, can you compare this particular structure with humans or any mammal? Yes. So these are the male gametes. So when, in case of humans, the sperms are produced, so there should be some tube which will carry these sperms to the vaginal tract or the reproduction site. Okay, we are not talking about how the sperms will travel. So simply think about, this is how the similarity is there between animals and the plants. Here the pollen tube is formed and it carries male gametes along with it. And in case of humans, penis is present and urethra, so through which the sperms they travel and they reach to the appropriate site. So this is how you can compare the things. So this is nothing but the development of overall pollen grain. Okay. Now we can consider a typical structure of female gametophyte. So you are well aware about the words ovule, then embryo, embryo sac, triploid endosperm, then antipodal cells, chalaza end, micropylon. There are so many terminologies, so you need not to worry about it. So we are going to learn each and everything in detail. Okay. This particular foundation course is just for revising the basic things which you have learned in the standard 8, 9 and 10. It is possible for many of you that some of you can enjoy this topic, some of you will feel bored, some of you will say अभी तो कुछ नहीं है, बाद में देखेंगे, उसका जब भी आएगा रेफरेंस तो वो टाइम पे पढ़ लेंगे, अभी क्या करना है इसका. So we need to study zoology more because we want to be a doctor. It is not the thing. Better you should study biology and the goal will be achieved. We assure you. Okay. So let us get back to the structure of female gametophyte. Do you remember this particular structure is a structure of ovule? This is much magnified structure. So this is a typical point where that wall was present. Do you remember? So this wall was present, similar to this. Okay. So this wall, it shows attachment of funicle, means a stalk. Funicle, a stalk. If you remember word funicle, fine. If you don't remember it, then you can simply call it as stalk. It's a stalk-like structure with which the entire ovule it is attached to the ovary wall. That's it. Now think about the actual structure of an ovule, how the ovule is present. So this particular type of ovule is called as anatropous ovule. Now you need to imagine the things. Consider this as a stigma, style and ovary. Consider this particular, my hand is ovary and this is the stigma disc which is present here. Then this is style and canal and this is ovary. Okay. Now we are considering this particular part, not this. So this particular structure, if it is inverted by position, not if it is not facing to the stigma disc, then this particular structure is called as anatropous type of ovule. There are so many details about types of ovule. We are not going to consider them right now for this particular foundation phase. But remember, this type of ovule is called as anatropous type of ovule. If it is placed in an upward direction, it is orthotropous. This is anatropous. So this anatropous ovule, first of all, let us think about the ends. There are two major ends. A fine pore which is present through which the pollen tube enters. इथे तयार झालेली आहे pollen tube, विसरायची नाही, कुठली pollen tube वगैरे असं म्हणायचं नाही. So this particular micropylon opening is a safe passage for entry of pollen tube. So this end is called as micropylon end. The end which is there opposite to the micropyle is called as chalaza end, because this region is called as chalaza, C-H-A-L-A-Z-A, chalaza. Chalaza end, micropylon end. There should be, so this particular structure is embryo sac. Now, if embryo sac is there, if egg cell is present, then there should be enough protection. To protect a developing embryo sac, there should be a presence of some jacket. So these jackets are nothing but the integuments. So this is outer integument and this is inner integument. So this is inner integument, right from this place to this place, and this is outer integument. Remember, this is ovule. When fertilization occurs, when development occurs, when proper growth occurs, that time ovule gets converted into seed. And if you remember, seed has a seed coat. Seed coat is made up of two layers, outer integument and lower integument, or you can say outer testa and tegmen. So these are the layers which are going to be there after seed formation. We are going to learn that. So this particular embryo sac is made up of several things. We are not going to learn about how embryo sac develops. So simply think about, this is anatropous ovule. So ovule is attached to the ovary wall with the help of funicle or a stalk. So these are the protective integuments. I can share a word with you that when two integuments are present, so it can be called as bitegmic. So this particular structure shows presence of two integuments. So tegma, layers. So these two integuments are present, hence it is called as bitegmic. Okay. Let us think about the main structure which takes place, which is actively participated in a reproduction or fertilization, you can say. So this is egg cell. Egg cell and these two sister cells are called as synergid cells. This egg cell plus synergid cell, collectively they are called as egg apparatus. I'll write a word, egg apparatus. So this egg apparatus is nothing but a three-cell structure. What three cells are there? Egg cell plus two synergid cells, egg apparatus. Think about and yes, egg cells are always present near the micropylar end, or very near to the micropylar opening, you can say. Then at the opposite end, at chalazar end, there is presence of antipodal cells. Antipodal cells are present in such a way that they provide some nourishment, they provide some additional support to the developing embryo. Okay. There are so many functions, but these functions we cannot consider in this particular course. Okay. So egg cell is always they are guarded by the sister cells, and these sister cells, they have filiform apparatus with them. This filiform apparatus, I have not shown that particular structure. It's a palm-like structure which is present on the typical sister cell or synergid cell, and this filiform apparatus, it helps in the proper welcoming of a pollen tube when it enters through the micropylar opening. Okay, so simply listen to these things, and if you can remember them, you can remember, otherwise you can forget them. So these are the synergid cells, egg cell, antipodal cells. Now think about this particular structure which is present in the middle. So these are not two cells, we can say. This is a cell with two nuclei. So this is secondary nucleus. Secondary nucleus is most important structure because it takes place or it performs a function of providing a nutrition to the developing embryo. How? We are going to learn that. Let us think about entry of this pollen tube through the micropylar end. Okay, now pollen tube has entered, so it will reach here, and these synergid cells where the filiform apparatus is present, So they will welcome the pollen tube, the male gamete actually. And during this journey, continuous travel of this pollen tube through styler canal, and when it reaches to the ovule, so that time we can expect this tube nucleus it degenerates. So whenever there is an entry of pollen tube through micropylar opening, there are two male gametes only. Two male gametes, they reach here, and first, which is first one, this one. So it fuses with the egg cell. So this egg cell and this first male gamete, it fuses to form a diploid zygote. Now, what about this second male gamete? बरोबर यायचे म्हणून आलेलं नाही. It's a specific plan of a nature. Once this particular structure has developed into a zygote, which will further convert into embryo. So this particular structure requires some strong nourishment, and that nourishment is provided by endosperm. So endosperm, which is triploid by nature, and it is formed by this particular second male gamete and these two secondary nuclei. So this secondary nucleus and the second male gamete, it forms a typical triploid structure which is called as primary endosperm nucleus, PEN. I can write a word here. Primary endosperm nucleus, PEN. Further, which divides mitotically, and it forms triploid endosperm. Don't worry about the ploidy level, what is ploidy level and other things. So these structures which are there, let there be egg cell or secondary nuclei, both of them they take place in reproduction, that is fertilization. The first male gamete fuses with egg cell, and second male gamete fuses with secondary nucleus. So this is how the things are completed. So this basic concept we have learned. Okay, I think we can switch to third board after a pause. Let us think about the post-fertilization changes. So these are the actual fertilization changes, and not only this particular structure, egg cell changes or secondary nuclei changes, everything changes. So there are so many changes, there are so many chemical reactions, or I can say there is a series of chemical reaction which is taking place during fertilization. And if you remember, you have seen the developed ovules, means seeds of okra or bhindi plant. Okay, so this is how the nature works in an excellent manner, and definitely there are certain flaws. Yes, some of you can recall the things. There were certain ovules which were unfertilized, so they remain smaller in the size, and they were placed on a central axis. If you remember it, if not, you can observe that, you can listen to that video once again. So definitely there are certain changes. Let us think about the structures which are formed or transformed, we can say. Overall, ovary. So ovary, it gets ripened. So it is developed. It is modified structure now after fertilization. So it gets modified into fruit. The fruit which we eat is nothing but ripened ovary. How it ripens? Just because of fertilization. Because embryo has formed, embryo is ready, embryo forms, further seed structure it shows, then the seed has to be there in a proper fleshy thing just to protect itself. Fruits are not meant for we people. Mango is not there for we people. हा छान लागतो आंबा खायचा आहे म्हणजे माणसाला आंबा दिलाच पाहिजे देवाने असं नाही आहे. So it is just about I'll show you yes, can you please get it? So I'll show you one typical example, and most of you have seen this. So it is not a God's gift to we people, it's a God's gift to the plant, just to survive in a nature in all harsh conditions. So this is how the structure is there, beautiful structure around this. This is mango seed. So around this a fleshy part was available and it was consumed by we people, and this is how after germination the new plant is growing. So always remember, this protection part is given to the in favor of plants only, not in favor of humans. We people try to consume the things, but we cannot expect that the things are there for we people. Let us think about the ovary once again. When ovary is ripened, so it gets transformed into fruit. Ovary wall, fruit wall. Okay, so it is nothing but the pericarp. Okay, then ovule, as I mentioned, this ovule after fertilization it forms a seed. Yes, always the seeds are present in a fruit. Then integuments are there, outer integument and inner integument. Bitegmic, if you remember the word, I have mentioned it. So bitegmic integuments means outer and inner. They get converted into testa and tegmen respectively. Okay. Now about secondary nucleus. It forms endosperm and the egg cell. After fertilization, it forms embryo. What about antipodal cells? So they get degenerated once the things are growing, once this particular mass is filled with a triploid endosperm, and if embryo grows in a particular direction, that time these antipodal cells they get degenerated. So this is how the things are there with respect to angiospermic development. Okay, I think now we can focus on development of a typical seed. So have you understood a concept of development of egg? Now egg has developed endosperm, which provides a typical nourishment for developing embryo. It has formed. It's a diploid endosperm. It divides mitotically, it divides rapidly, and it forms a typical bunch full of food, ready food available for developing embryo. So you can consider this particular event with us. In case of humans, when baby is there in a mother's womb, mother's uterus, so that time this particular developing embryo, it receives food from the mother, from mother itself. So the umbilical cord, or there are certain we can say there are certain patches you have learnt about that provides a nutrition to the developing embryo. You can consider this particular event, fertilization event, with humans fertilization event. When pollen tube enters into the embryo sac or up to the embryo sac, so the first male gamete fuses with the egg cell. In case of humans, eggs are produced by the ovary and it is taken to the fallopian tube. Sperm travels through the vaginal canal and it travels, the sperm cells actually they travel up to the fallopian tubes. Now once the sperm cells they have reached to the desired site, that time the very first sperm cell it touches to the wall of the egg and the fusion occurs, and this is how the sperm head enters and the further processes are there. You can compare that event with that of angiospermic plants. Even here, when tube enters through the micropylor opening, when it touches to the wall of a embryo site, so the first male gamete, it immediately fuses with this particular egg cell. The second male gamete, it enters and it reaches to the secondary nucleus, then it fuses. Here in case of humans, only one sperm, it fertilizes a single egg. If you remember, it was told to you in a human reproduction chapter in this particular foundation phase that every month only one egg is produced. Suppose this month, in the month of June, egg is produced from left ovary. Then in the month of July, the one egg will be produced from right ovary. So left, right. So this alternative cycle will be there. But every month, a single egg, ovum, will be produced. Okay. So this is the major comparison. Let us focus on the development of dicot seed and monocot seed. Dicot means two cotyledons are present, monocot single cotyledon present. Dicot seeds are called as non-endospermic seeds and monocotyledons are called as endospermic seeds. These particular details are already there in your standard 8, 9, 10 books, and surely in your regular curriculum we are going to learn each and everything related to these terminologies. Okay, let us revise the things. Some of you will say what this butterfly is doing here. It is not a butterfly. It is simply an embryo axis. So this is embryo axis. And these are the two cotyledons. Yes, we are going to have a demo for this. Once we finish with this part, we are going to have a demo for this. And always remember, whenever we here at Learning Center will share a knowledge with you, we'll always try to share a demo, we'll always try to share a practical, or we'll always try to share at least a photo for the same. Because these are the two-dimensional structures, these are hand-drawn structures. But whenever you observe a real plant or a real sample, that time you can remember the things in a better way. So this is just to make you aware about what type of knowledge is available, what type of magic is created by the nature. So this is a cotyledon. So these are the two cotyledons. So they are sealed like this, and if you germinate, if you put some seeds in the water overnight, if you put some gram seeds in the water, then definitely they will swell up. And if you remove them from water, and if you tie them with a cotton, or if you put them in a some incubation type of chamber, then definitely they will germinate and they will produce a radical first. So that particular structure will be radical. So it will get first its entry into the soil, it will establish itself, and then the plumule, which develops as a shoot part, it will develop. So plumule and radical both are there. So this part is nothing but the radical. So this part is upper part, which will be forming a shoot, that is plumule. So this cotyledon 1 and cotyledon 2, they provide a nourishment to the developing embryo. So this is once again a point for comparison. In case of humans or in case of birds, we can compare. Yolk is present. So yolk is present just for development of, it's a food for developing baby. Similar to that condition, the angiospermic plants or plant, they always provide a cotyledon which are full of some nutritive food. So they are always taken up by the developing embryo. So this is embryo axis. So this part, which is this particular cotyledons are attached, so it is called as first node. Always remember, we are well aware about node and internodes. The node is a part at which a typical leaf is attached, that is a nodal junction, and the part which is present between two nodes is called as internodes. Consider, so these are the nodes and this is internode. Okay. Now think about this typical structure. The part which is below first node is called as hypocotyle, hypo below, and the part which is above the first node is called as epicotyle, above. Okay, epicotyle and hypocotyle. This is embryo axis, this is radical. Radical first comes out because it has to establish itself into a soil to form a root system. Once the root system is set, immediately the shoot system will grow, and shoot system will grow because of plumule. Plumule will develop and it will form a typical leaf system, shoot system, and photosynthesis will start. But till then, all the necessary support of food will be given by these cotyledons. So these cotyledons, they contain the endosperm stored within them. So endosperm is already absorbed by these cotyledons. So always remember, in case of dicots, free endosperm is not available. Now let us think about the monocots, how the things are present in monocots. So can you observe this diagram? It's a typical maize grain. Once again, it's a vertical section. Vertical section, it shows presence of ample of endosperm, free endosperm. Starch is freely available. आपण हे करतो ना, मक्याचं पीठ असतं, कॉर्नफ्लोअर वगैरे. So it is nothing but the starch. The Chinese मध्ये use करतात just to get a thickness to the gravy. Okay. So this endosperm is nothing but a typical stored food material for developing embryo. We are not going to get ourselves confused with all the terminologies, but simply we can say this is developing part is plumule and this part is radical. Now there should be some jacketing. There should be some jacketing around each developing part. So if this plumule is going to be there as a primitive phase for shoot system, and if radical is going to be there as a primitive phase for root system, then those systems should be protected well. No infection should be there, no bacterial attack should be there. So this particular structure, plumule, is covered by coleoptile, and this radical is covered by coleorhiza, rhizoids, roots. So these are the terminologies which you need to remember. Biology is all about terminologies. Physics is all about technical subject, technical knowledge and application of your intelligence. Chemistry is, we can say, about understanding how the reaction occurs, how molecules are transferred from one place to another place. That is all about chemistry. But here, in case of biology, or botany, zoology, anything, you need to remember the terminologies. So they have maximum words are there from Greek language. So they have the Greek origin, and there are some good terminologies which are said by those Greek people. So it is our duty to understand the things well from biology and to study them. Okay. After a pause, we'll continue with types of germinations. So this is a typical. This is the board for germination. Let us think about, there are two major types of germinations. Okay, first is hypogeal type of germination, and second is epigeal. Hypo means below, epi means above. So hypo, below the soil, and epi means above the soil. So what goes below the soil and what comes above the soil? So that is seed or cotyledon. Don't think about the monocot or dicot. Okay, Consider this particular set of diagram. This particular seed remains there below the soil even if the plant has grown into root and shoot. Now this complete plant has grown, tall plant with some root system, supportive root system, and even then you cannot say that this particular seed has come out from the soil and it is growing in an upward direction. It is not a case. But here in case of epigeal germination, epigeal specifically we can say a bean seed. If you sow it after some time, or we can say in a day or two, a radical will grow. Intermediate stages we are not showing here, and after a week or so the radical will develop more into a proper root system. These roots will establish itself first, and the seed, or you can say the cotyledon which is there, it will try to come out. The cotyledon will try to come out from a soil. Here I can show you the things. The cotyledon is trying to move upward. In next stage, it will move more out, and then immediately after getting out from a soil, this particular plant will grow, and the cotyledons, both which are here, they will share all their food with developing plant. So this particular series of diagrams is just for your understanding. These are not the exact stages, but yes, this is the sequence. So here you can say that this wrinkled the shrunken structure of a seed is just because of its old age, one sense you can say, because this particular structure, it provides a food material to the developing shoot, that is plumule. We have learnt about this. So the plumule develops into shoot, and radical develops into root, and this grows first, this grows later. Remember, these are the simple things. So once again, in hypogeal type of germination, the germination which occurs below the soil, and even after the formation of radical and growth of radical and plumule, the seed remains below the soil level. So this particular example can be studied well with the help of maize seeds. So if you sow a seed, then it will form plumule and radical. Radical will grow first, it will establish itself, then plumule will grow, and this is how the monocot plant will be formed. So this is a usual case of monocot, that is hypogeal type of germination. Here in case of epigeal germination, we have considered a bean seed. We are going to show you a bean seed. So this particular bean was put in the soil. Then this particular structure, it has started producing a radical, a typical structure which will further develop into a shoot system, a root system, and this particular cotyledon part will be pushed away from a soil, from the soil to the outside, and it has come out, and now this plant has established itself. So this is how a typical epigeal and hypogeal type of germination can be seen. Here you can consider endosperm was present, free endosperm. We have seen this. So free endosperm was present, and now it has been utilized by a developing plant, but still the seed remains in the soil. Here in case of bean seeds, the endosperm was trapped in a cotyledon, but it was consumed by a developing plant, developing embryo. Okay, so now it has helped a plant to develop into a fully developed shoot system and fully developed root system. So these are the two major types. The third part is there we are going to consider is a typical vivipary. So what is vivipary germination? So is this title visible? Okay. So vivipary, giving birth to young ones. What is ovipary? Giving birth to young ones, but the intermediate stage is egg stage. So eggs are given out and the development occurs in the egg. The yolk is present just to fulfill the nutrition requirements, and then the young one comes out. Okay, but here in case of vivipary, in case of animals we can understand the things very well, but how is it possible to consider a viviparity in plants? So it's a once again excellent creation of nature. Example, Rhizophora soneracea. Don't worry about the examples. Kharfuti चांगलं माहिती आहे, or mangroves. So these are the mangroves. Soneracea, then Rhizophora. There are so many n number of species. Can you think about a plant, any plant, say a tulsi plant or mango plant, if you put it in a salt conditions, or if you put it in a what we can say salt water or a seashore, immediately it will die because there will be maximum uptake of minerals, and if minerals are taken more, then definitely there will be a process of exosmosis. The water will be given out and plant will dry immediately, the cells will dry. But here the plants have adapted themselves to survive in a saline conditions. So these plants are called as halophytes. Halophytes means the plants which love saline conditions, salty conditions. So these are halophytes. So entire plant we are not able to show here, but we have a picture and during demo I'll show you. So consider a typical branch which is present. There are so many branches you can imagine. A typical branch, a flower which further develops into a structure which is called as a fruit initially. So this particular structure is present like this only, a corolla and a fruit. So this is fruit is nothing but a developed ovary. If it is ovary, then definitely there should be seeds, and this viviparity is best example of development of seed, germination of seed in the fruit itself. So one of the seeds, so it has come out and it has formed a radicle. It forms a dart-like structure. So this is dart, and if suppose if it is falling down in a muddy soil, so it will get fixed somewhere in a soil like this. So it will get fixed. So this is how this radicle grows. This is hypocotyle, developed hypocotyle. So this falls from a tree and it establishes itself in a muddy soil. So you cannot say that this particular plant is present somewhere in the middle of a sea. So it is not a thing. So it is present in this on the seashore. Okay. So definitely whatever the branches are there, so they will be hanging out on the muddy soil itself. So this particular structure, this dart-like structure, will get fixed in a soil, and each structure will develop itself into a new plant. So this is viviparity. Once again, we will consider a thing. Consider this as a branch. So this branch, it produces flowers. Flower when it get fertilized or pollinated. So that time there is a fruit formation. Fruit formation means there is a seed setting already. So this seed will not come out. Seed will not come out and then it will not germinate. Seed will germinate itself in a fruit. or the cavity of a fruit, and it will form a radicle. So this radicle further it will grow, it will form a hypocotyle, and this dart-like structure will fall down from a tree, and it will get fixed in a soil. Sometimes it may happen that this particular structure will fall down, but it will not get fixed in a soil. So sometimes it may fall down and it can flow along with a water current, and at a particular place it will get fixed and it will form a root system. Vertical growth of plant will occur. So this is a typical development or evolution or beauty of nature which we are studying. Okay, so I think after this we will switch to the demo table. So this particular structure of a seed, if you observe carefully, this is a dicot seed. Okay, so seed coat, testamen which I have mentioned earlier. Then this is embryo axis, and it can be given so many names to different parts. So embryonic leaves, the initial leaf primordial, which we can call it as. So these are the leaves. Then the embryonic foot. Then further it develops and forms a root system. This is embryonic shoot. Further it will develop and it will form a separate shoot system, and a huge tree can be formed out of a small seed. Okay, so after this we can switch to the another image, that is structure of typical seed. Once again, this is radicle, this is plumule, these are the cotyledons, and this is the first node. Better concepts can be cleared with this picture. So this is a simple embryonal axis. Imagine these are the cotyledons which are present here on this side, either side. This is plumule, this is epicotyle, this is first node, this is hypocotyle, and this is radicle. Suppose the radical region starts from here, from this part. So the region between first node and radicle is called as hypo, means below, hypocotyle. This is plumule part starts from here, and the region between first node and plumule is called as epicotyle, above. Okay. So after this particular dicot structure, we can consider this monocot structure. Once again, we have observed this diagram on the board, so don't go much into the details. So simply think about the typical structure of a monocot seed, that is maize seed, pericarp, that is outer covering. Now let me tell you one thing. So this particular maize or jowar or any grain, wheat, so all these are fruits. It's a single seeded fruit. Hence, when testamen, they are fused from outside, and when they form a pericarp, so you cannot say that this particular structure is seed and that particular structure was a fruit. So in this particular case, in this wheat or jowar or bajra or maize, you cannot say that it's a seed only. It's a single seeded fruit. Okay. This particular part is filled with endosperm, excellent provision for developing new one. So this particular part grows into plumule, and plumule is covered by coleoptile. Similarly, the radical part, it will grow into a root system, and it is covered by coleorhiza. So this is a typical seed structure for monocot. Okay, then think about a fruit. This is a mango fruit. Obviously, this is a stalk. If you remember, I had mentioned about funicle. Funicle is nothing but a stalk-like structure. So this is a stalk-like structure. Now this particular structure is ovary. Are you getting my point? This particular structure is ovary, and the seed which is placed here is nothing but the ovule now which is fertilized and which is matured to give rise to another plant is called as seed. So this is seed covered by pericarp. So pericarp further it can be divided into epicarp, mesocarp and endocarp. Epi outer, meso middle, endo the innermost. Immediately next to the seed or seed coat, the endocarp is present. Endocarp is covered by the mesocarp and the mesocarp is covered by skinny epicarp. Now let me ask you a question about this particular structure. So this is a mango fruit. Okay. So which part do we consume? It's a sweet, tasty, fragrance, fantastic aroma. So which part we consume? So this particular mesocarp, yes, mesocarp we consume. And have you observed when you cut a mango with the help of knife? Have you observed that particular mesocarp? It may contain some fibers. I'm not talking about the improved variety of any plant, but still there are fibers which are easily observable, and you can test it. You can feel it while eating mangoes. So this particular part is filled with fibers and fleshy juices, and it is covered and protected by skinny epicarp. So this is how a typical fruit develops from an ovary. You can compare these structures. Once again, seed. So this is coconut and this is mango. This is seed of mango. This is you cannot say this particular structure hollow part is seed. It is not a seed. It's a hollow part which is filled with liquid endosperm. Okay. Yes, it is mentioned here, liquid endosperm. Then this particular white part, cannon, which we eat regularly. So that part, white part, is solid endosperm, a regular endosperm, and the liquid which is present inside the cavity. So it is liquid endosperm, and it is equally nutritive by its nature. So solid plus liquid endosperm, they are present, and these are the mesocarp region. And yes, if you remember, mesocarp is very fibrous. So these are nothing but the mesocarp, sclerenchymatous. Yes, some of you are saying correctly. So this is sclerenchymatous tissues. This stony part is endocarp. Let me tell you, even in case of mangoes, yes, we can have an example now. Even in case of mango, so this particular part is stony by nature. So this endocarp is nothing but the protection which is present for this particular developing structure. Okay. Similar to this, नारळामध्ये जी करवंटी असते ना, कडक येतं का लक्षात, फोडताना जे ज्याच्यावरती काहीतरी ब्रेक करावं लागतं. सो दॅट पर्टिक्युलर रिजन is nothing but the endocarp, stony endocarp, and then the outer very thin skin is available. Initially in the fresh coconut it appears to be green, a smooth silky green. Okay, it's a very thin papery cover, and as this particular fruit, this coconut dries, so it turns into light brown or dark brown depending on the composition of the skin. So this is how the fruit is developed, and this is a comparative study of fruits. So this is fleshy fruit and this is non-fleshy fruit, you can say. Let us think about now the germination. Now you can tell me, what type of germination is this? Epigeal or hypogeal? Hypogeal. Yes, this is hypogeal because the seed which is present here, it remains below the soil even if the plant is growing. So this is a typical case of hypogeal type of germination. Now consider this particular plant. So even if the plant is growing, even this particular mango plant is dicot, still this particular structure, this particular seed remains below the soil. So this particular case is of hypogeal type of germination. Okay, now we are going to use this particular plant for many times. Let us think about the apigeal type of germination. What is apigeal? So the bean seeds are there, and yes, they come out when the coleoptile covered plumule is coming out, and when it forms a separate shoot system, that time this particular cotyledons, they also travel upward above the soil, they come out from the soil and they grow, and as this particular plant develops, ultimately they wither off, they fall down, whatever the content is there, endospermic content is there, so it is utilized by the developing plant. Once the leaf system is set, so there is no need of cotyledons anymore. So the attachment point of these cotyledons is called as first node. Do you remember embryo axis? The first node where the cotyledons were attached. So this is the first point. And sometimes when these cotyledons they come up, they turn into green color. So I am going to show you. This green color, it appears there just for photosynthesis. A small amount of photosynthesis process is carried out by these green cotyledons, and yes, these green cotyledons they support this newly growing plant, and once this particular set of leaves are formed, once this system is set, so they wither off, they fall down, and this plant grow rapidly with several mitotic divisions and so on. So let us get back to the germination part first. We'll finish with the photographs first. So this particular plant you can remember. So this is Rhizophora. This is mangrove plant. Another word for this particular plant is Kharafutichi vana kiva Kandal vana. Okay. So there are so many Marathi terminologies available, but popularly it is called as mangrove plants. And yes, look at the plant. So that action has occurred, and immediately after that this particular plant starts growing. You can observe the same phenomenon in a sketched form. So this structure was shown to you on a board. So this inflorescence, so it develops a fruit, then radical comes out because seed germinates inside the fruit. So this is viviparity. So this is how radical comes out, and this is hypocotyle, and because this particular part is between first node and the radical part, so this particular intermediate part is called as hypocotyle. So this is how it falls down in a muddy region or a swampy area, and this is how it forms a separate root system for it, and remaining part grows above the water level, and it forms a shoot system, and further photosynthesis process occurs as usual. So I think we have done with this particular part. Now let me tell you few things about this. Yes, we can now have a close look. Consider this particular part was, so better it can be demonstrated with forceps. So this was the plant, and by mistake I detached it from the root system. So this is how the plant is growing. So this is a grown stage. Let us start with this particular gram slave. Okay. Now, focus. Let me tell you this particular point you need to remember, through which the radicle is coming out. Radicle first comes out. It forms a root system, and this radicle is solely responsible for forming well-developed root system, because nutrition and I can say more than nutrition, minerals are required. Minerals and capillary water is absorbed by the root hairs. So this is how the radicle is useful, and this radicle comes out from a pore, and this pore is nothing but the remnant of micropylar opening. Do you remember? Yes. So micropylar opening through which the pollen tube enters, and it is the same thing through which radicle comes out. It's a fantastic design by nature, and we people always keep on admiring human race only, that yes, we have done these things and that thing and we are so developed. So it is not like that. So always keep some faith in nature. So I am removing a typical seed coat. Now tell me what I am removing exactly. Yes, some of you are saying it correctly. I am removing intestine tegmen. Some are saying yes, you are removing developed integuments. Yes, it's a bitegmic structure, or it was a bitegmic structure. Tegma means layers. So two layers were there, inner and outer integuments. Now outer integument has converted itself into testa, and inner integument has converted itself into tegmen. So focus on this. Let me tell you certain things very clearly. So if you observe this, you can easily say this particular structure is nothing but the seed, and this particular structure is nothing but the seed coat. But where are the testa and tegmen, two separate layers? Well, this particular outer hard part is nothing but the testa, and the inner part, which is shiny, lustrous, which is fused with the outer part, is tegmen. It's a peppery structure. Okay, so this is how we are observing the things, and let me tell you whether embryo is present or not. Yes, it is present. Can you observe this? I'll hold the things like this, and yes, some of you are pointing out the things very clearly. Can we zoom it? Is it possible? Yes, thank you. So this particular structure, focus on this. So this structure is nothing but the developing plumule. So this entire structure is called as embryo axis, right from this tip to this curved tip. It is not actually curved, it is just because of its natural way to find out the soil to germinate. So this is how the structures are there. So this is embryo axis, plumule, radicle, hypocotyle, epicotyle, and the part where the cotyledons are attached. So this is first node. Let me tell you, this particular botany part is quite interesting, and you can perform these experiments at home. So you can take bigger seeds and you can soak them overnight and put them in a chamber where they can form radicle, means they can germinate. So this is how you can easily observe, and if you have lens or something else, you can observe the things under it, or else you can take a photograph out of it and then you can zoom it and observe the things. Okay, so this particular structure, I'll remove one cotyledon. Yes, so this is the first node. Are you getting my point? So this is the first node, this is plumule, radical, first node, hypocotyle, epicotyle, everything you can observe. The nice things are there. Let me dissect the another one. So it is always joyful to handle plants. It is always joyful to handle various section practicals or dissections. So always remember, biology cannot learn without the experiments. And just because of this pandemic situation, you are not able to come here, but still we are trying to show you maximum things as and when possible. Yes, Kaustubh sir, can you please focus on this, and I can easily observe the things are quite green in color. Yes, thank you. So this particular structure develops further into a shoot. Yes, green part you can observe. This is plumule. Diagram मध्ये आपण कसं काढतो, इथे कसं दिसतं. So always remember, diagrams are nothing but the representative pictures of the structures which are there actually in the nature. So always try to remember the original things. So this is another thing. Let us have a look at this particular structure now. Let me tell you in the beginning itself. This particular seed was gram seed, and gram seed, the adjacent to gram seed is a germinated mung seed. Okay, so this is not a gram seed. Let me tell you in the beginning. Now you can observe how the things are developed. So you can easily say whether this particular structure is epicotyle or hypocotyle. What is this? So this structure is epicotyle because root system is here and everything is growing above the soil level. Do you remember the diagram? If not, you can go back and you can rewind the video and you can easily watch the things. So these are the shoot systems. Yes, the beautiful things are there in the nature, and yes, we still keep on saying that there is nothing to do in the lockdown period and we have nothing to play, we have nothing to do. So hobbies you can develop, or you can start with new hobbies like gardening or dissecting plants or doing something else. So this is how the things are there. One more thing I'll show you. So this is a typical mango plant, the giant one. Once again, let me remind you one thing, that this particular structure is a hard stony structure. Even with the help of my hand I cannot remove them. I'll still try, but I'm not sure whether I'm able to do it or not. With the help of some instruments I'll try to pull out the things. No, still it is not possible. It is too hard, and it is too no, it is not possible for me to separate out this innermost part of this seed, otherwise this particular structure will get disturbed. Yes. Beautiful. Fantastic. Look at the seed. आंब्याची कोय म्हणतो ना आपण. So this is nothing but the actually seed. It's a dicot structure. Let me show you a dicot structure with this dissected try to observe the things which are there. So this is a hypocotyle. Hypocotyle means this particular hypogeal type of germination, means this particular seed part, it never comes out of the soil. So this part grows up. This remains in the soil. This is how the things grow. So this is the basic structure of growth for a mango plant. Yes, you can observe. So this is the first node we can easily trace out. Oh, sorry, my mistake. I removed one cotyledon. So this particular structure is nothing but the you can observe all these fleshy mass is nothing but a typical endosperm, or you can say it's a cotyledon, but endosperm is being trapped in it. Okay, so this is how the structure appears. Okay. Let me tell you another fact about this particular plant. I think we'll keep it aside. Now observe this particular plant. This is, yes, come on, say it. This is apigeal type of germination. Yes, these are the cotyledons. These are the cotyledons. These are the cotyledons. So, sorry, once again. So this is how they wither off. So these are the cotyledons. You can easily observe the cotyledon color has changed to green. So this particular seed was, or this particular plant is of rajma. So rajma was soaked overnight and then it was put in a germination chamber. And once the germination has occurred, we put it into a soil and then it started growing as a individual plant. So this particular cotyledon, initially it was not green, now it is green, just because it's a apigeal type of germination. It has come out and now it is showing its responsibility to provide a food to developing plant, and also it should convert itself into a green structure just to support, to carry out additional photosynthesis. So this is a typical structure. You can observe closely, you can observe. Yes, are you able to focus this? Yes, great. Okay, now another plant is a gram plant, for which a gram seed was shown to you, and these are the gram plants. Something else is also growing with these gram plants, but the small green leaves are of gram plants. This is hypogeal type of germination. You can observe. So no seed is traveling upward along with the growing plumule. So only shoot system is developing from the plumule, and remaining the seed part and the remaining part, it stays there below the soil. So this is how the apigeal, hypogeal and viviparous type of germination occurs in the nature. This particular part, this reproduction in angiosperms and some part of the growth is now completed. So you will be given certain study material along with the worksheets, which you need to solve in your regular notebook. And well, I think you should remember the terminologies only. Don't force yourself to remember each and everything which is said here on the stage. So it is always better for you people to understand the concept. You can simply correlate the botany concepts with zoology concepts. So it is possible. Even as as we do it in class sessions or regular 8, 9, 10 standard, compare this with that. So not only compare, you can you should not write the differences. You can compare, means actually you should compare the similarities. So how this particular structure resembles with the another structure. So this will be the exact real way of learning science. I think with this we have done with that chapter. Now there are certain instructions about various issues. First of all, after watching this video, immediately you can talk to your parents about continuation of standard 11 by online way. If you have talked to your parents, if you have seen video, if you have filled the form, then forget this message. You need not to send twice or thrice confirmation forms to us. A single form will suffice. 11वी करताच आहे तुम्ही 11वी 12वी फर्स्ट इयर सेकंड इयर सगळ्याचं ऑनलाईनचं कन्फर्मेशन आता देऊ नका. So simply this form will be there available, a link will be there and you need to click it, you need to fill it whether you are interested, whether you are willing to continue with online method of learning or not. Simply mention yes, click the yes button. If no, say no and submit it to us. So this will help us to take a judgment for launching a particular learning app, because we have done with the app, we have developed it, and once we get a confirmation, so those who have confirmed their admissions, they will get access to the study material through app. Currently we are using Google Classroom, and this particular method, app method, will be implemented for regular standard 11 chapters. Next instruction about this particular app technology is in a day or two we'll provide you another form. I know you people will get confused. Just to avoid this confusion, immediately finish with the first task. What is first task? So your first task is to give confirmation whether you want to continue with online method for entire standard 11 or not. Yesterday we have posted a video. Along with that we have sent one notice, Marathi notice as well as English notice. Show it to your parents, discuss with your parents, and then decide and then send your inclination form to us. Once you are done with this first task, second task is for filling a form for your contact numbers. So this particular app will be operative with the help of your mobile numbers. We are doing all these things well in advance because we know that some people may change their mobile numbers due to some emergencies, due to some problems. They may have to change their regular contact numbers. So let me tell you in the beginning itself, students and parents, both of them, they should submit their phone numbers which are fixed with them, which will be there with them for entire year. I repeat my statement. तुम्ही तुमचे जे फोन नंबर्स पूर्ण वर्षभर वापरणार आहात तेच फोन नंबर्स आम्हाला जो आता तुम्हाला फॉर्म पाठवला जाईल त्यावरती लिहून पाठवायचे. So this particular phone number will help us to keep you in a loop of application mode. Okay? तुम्ही जर का तो फोन नंबर मध्येच बदलला तुम्हाला ऍक्सेस मिळणार नाही. So you should stick to one number only. ज्या पेरेंट्सकडे ज्या स्टुडंट्सकडे एकच नंबर आहे आणि पेरेंट्सचाच नंबर ते यूज करतायत, शक्य असेल तर त्यांनी स्वतःचा नंबर घ्यावा. Because there are certain instructions which can be sent directly to the parents. Okay? So this is in your favor. It is not a typical track for you people to trace whether you people are working or not. It is not a thing. Simply take a separate number for student, take a separate number for parents, and keep these numbers at least for 11th standard. And these numbers should be constant. You cannot change it. Once you change it, we cannot do anything because the system operation end is at the agency side. We cannot do each and everything, each and every single operation in this center. Learning biology is easy for we people. This technology part we have to do in cooperation with you. We cannot do the things in at our end. Okay. So तुम्ही एक लक्षात घ्या की हा जो काही टेक्नॉलॉजी पार्ट आहे तो तुम्हाला नवीन आहे, आम्हालाही नवीन आहे. ॲप्लिकेशन तुम्हालाही नवीन आहे. खूप लोकांना गुगल क्लासरूमचेच प्रॉब्लेम्स अजून येत आहेत. त्यामुळे ॲप्लिकेशन जर का तुम्हाला स्मूथ हवा असेल तर तुमचा एक कॉन्टॅक्ट नंबर स्टुडंटचा, एक कॉन्टॅक्ट नंबर पेरेंट्सचा, एवढे 2 कॉन्टॅक्ट नंबर त्या फॉर्ममध्ये द्यायचे. आता फॉर्म कधी दिला जाईल? when this form will be posted. हा फॉर्म तुम्हाला येत्या 1-2 दिवसामध्ये मिळेल. त्यामुळे तुम्ही कोणी फोन करू नका की हा फॉर्म कधी मिळेल. आम्ही तुम्हाला देऊ. आम्हाला अंदाज आला की याला फॉर्म द्यायला पाहिजे त्यावेळेस हा फॉर्म दिला जाईल. So you people should not call us when this form will be posted, when we'll get a link to fill the form. So don't do it. We'll post the link, we'll post a form at appropriate time. Okay? I think after this chapter we are going to start with plant kingdom and diversity of plants. Okay? So I think you can study these contents from your 8, 9, 10 board books or any board book. And you can get familiar with the various terminologies. Okay? Till then, goodbye.